Oh, okay.
You see this?
Oh, okay.
What is it? 58. 2 minutes.
Hi Chetan, can you hear me?
Hey, yeah, I can hear you.
How many people do you have on your center?
Right now it's just me and Chetan.
I don't know, and Jane haven't, we don't understand you, Jane.
12:20 also,
I
right?
know, but I don't know whether she's going for just 20 minutes.
Well,
I think we should get started.
but all the, have they,
Did you email the Zoom link also to the
Yes, I
pharmacy?
emailed the Zoom.
Yeah, I'm
because
not
that's,
sure where
this is
they
a
are.
requirement.
I think I emailed to everyone that they should be here 24/7.
Yeah,
Maybe they're
and
gonna
I
just
also
get on the-
mentioned that on the email as well.
Yeah, okay, thank you.
Dr. O'Donnell joined as well.
Yeah, Lauren's not feeling well, so that makes sense. So just gonna wait for Jane and the pharmacy students maybe on it.
I wanna be respectful of everyone's time.
Yeah.
Normally related to the 1 hour. but-
Yeah, let's get started.
Recording in progress.
Should I dim the lights?
That's a good idea.
Welcome everyone to my second seminar presentation. Today my topic's title is Assessing the Role and Mechanism Underlying the Melatonin-Mediated Actions on Muscle and Bone to Prevent or Treat Musculoskeletal Disease. I discussed my background in depth in my last fall seminar, but today I will just touch the key points that will be necessary to understand the rationale behind my aims and the assays I will perform. So before diving into what I did last year, I will just go through the background a bit. So let's start. Okay, the disease model or the disease I will specifically focus on is musculoskeletal disorder and the muscle part of it. So musculoskeletal system is composed of a bone, muscle and connective tissues, and its disorder is related to those tissues. It's mainly manifest as pain, risk of fall, imbalance and all that. So it's a very prevalent disease among elderly, especially in high-income countries, and it causes a lot of physical pain, and it is an expensive disease which also altogether causes mental health stress in the patient as well. What's alarming is due to stress, sedentary lifestyle and bad diet, people in their 40s are starting to manifesting symptoms of musculoskeletal disorder, especially osteopenia, osteoporosis and sarcopenia. Approximately 1.7 million people are affected yearly, and the total cost of musculoskeletal disease is $45 to $54 billion. So it has been affecting a lot of people. I have listed down the disease that falls under the umbrella term of musculoskeletal disorder. This side is bone related and that is muscle related. The most common disease bone related is osteopenia and osteoporosis, and muscle related is sarcopenia, which will be a major focus of my project. As I mentioned, that musculoskeletal disorder, it is a disorder or disease of muscle, bone and its connective tissue, mainly muscle and bone. Muscle and bone forms a single functional unit. They are very closely related, and disorder of muscle causes risk of fall, and thus in turns it causes risk of fracture. So as you can see, these are very closely interrelated. The cause of musculoskeletal disorder, or the factors that causes musculoskeletal disorder, are also common for both bone and muscle as well. The factors are mentioned here. Other than age, as you can see, inflammation, physical inactivity, fat infiltration, low protein intake, all kind of fades into muscle and bone loss in elderly and also right now in adults. Muscle and bone shares very similar paracrine and endocrine pathways, so the factors that causes bone loss also somehow factors in affects muscles as well. Since they share common anatomic, biochemical and cellular neighborhood, the factors that are affecting here in bone are also kind of relating back to muscle as well. One of the common thing that happens as people age is fat infiltration in both muscle and bones. Those fat cells produce adipokines that in turn destroy the formation of myocytes and osteocytes. Other than adipokines, myokines produced by muscle and osteokines produced by bones also attack each other. So together with age and fat infiltration, the health of musculoskeletal disorder deteriorates. These are some common osteokines and myokines that I have mentioned here. Since I will look into the muscle portion of muscular disorder more, I would like to go briefly about the role of skeletal muscle and what happens when sarcopenia occurs. So tissues of skeletal muscles are very important not only for our voluntary movement and posture control, they are also responsible for thermal regulation and maintaining the glucose uptake of the body. Any kind of disorder of muscle not only will cause you fall or impaired movement, it will also relate back to metabolic disorder like obesity and diabetes. The most common muscle-related disorder is sarcopenia. Here I have shown you a figure of a healthy muscle versus a sarcopenic muscle. With age, the muscle mass is shrink, and the fat, intramuscular and intermuscular, both fat infiltration gets higher. And those fat release cytokines like TNF-alpha, which causes the calcium-ion imbalance in the muscle. And calcium ion is very important for the excitation contraction function of the muscle. So when fat disrupts the calcium ion balance, the excitation contraction function is disrupted. As a result, people are more at risk of falls. If you go into the more molecular level, like what happened during sarcopenia, in this figure, as you can see, that aging that I have mentioned is one of the major risk factor. Other than aging, obesity kind of mimics the same thing that happens in the elderly. So this causes sarcopenia. The main two factors that is responsible for sarcopenia is inflammation and fat infiltration. With age and in obese patient, the inflammation rate gets higher. Like with aging, if I focus on that mainly, with aging the chronic inflammation rate is higher, and those inflammatory markers like IL-1 beta, TNF-alpha, IL-6, they cause metabolic disruption of lipid and adipocyte. So what happens is this inflammation cause more inflammation marker and also cause disruption in lipid metabolism. What happens when lipid metabolism is disrupted? The normal function of adipocyte or lipid metabolism is to store the excess fat as an inert lipid, excess energy source as an inert lipid vehicle. when their disruption occurs, this fat kind of spilled all over the peripheral areas of the body. And similarly, in the inter and intramuscle fat increase. And when all of this is happening, those inflammatory marker also causes muscle protein, the balance between muscle protein breakdown and muscle protein synthesis disruption. So that also not only increase the amount of lipid in the muscle, but also decrease the amount of muscle is producing in human. So these all together factor into sarcopenia. So the major takeaway is there that the reason of sarcopenia is two: inflammation and lipid infiltration in the muscle. So as I have mentioned, that aging is one of the major risk factor of sarcopenia, but sarcopenia is not the only thing that happens as people age. Here you can see the nocturnal melatonin level that also decrease as what people is aging. Here in the figure you can see the peak melatonin level is gradually decreasing as a person is getting older. So melatonin is initially thought to be the circadian rhythm controller, but with time we figured out that it does a lot of function physiologically, namely cardioprotection, neuroprotection, and even tumor suppression. For my project, I will specifically focus on the anti-inflammatory portion of melatonin by measuring inflammatory markers, and I will also look into the suppression of mitochondrial dysfunction, what melatonin does to mitochondrial function, and lastly the modulation of SIRT-wing, protein family that is closely related to inflammation and melatonin. So in my project, I will mainly try to tie the effect of melatonin and muscle and how it can play a beneficial role in sarcopenia. So before diving into that, I want to discuss what we already know about melatonin and its function in bone, so that my foundation of my project will be more clear to you guys. So first, on this side, it's a mechanism of action of melatonin on mesenchymal stem cells, like how it converts the differentiation of mesenchymal stem cell to osteoclast. This is extensive work done by a past graduate of our lab, Dr. Fatima Myount. So let me just walk you through it. So when melatonin binds to NT2 receptor, it initiates two signaling pathways. One is MAC1-2 and one is MAC5. Upon initiating MAC5 through ARC5, it increases the osteogenic protein like BMP2, FRA1, and RANKS2. These proteins are responsible for formation of osteoblasts. Similarly, MAC1-2 and ARC1-2 pathway increase the level of those same three proteins. But what MAC1-2 ARC1-2 pathway additionally does is it inhibits the PPAR gamma activity. PPAR gamma is mainly responsible for adipocyte formation and lipid metabolism in the body. So when MAC1-2 ARC1-2 path is activated, it lowers the PPAR gamma level or inhibits the PPAR gamma activity. So the differentiation of mesenchymal stem cells from adipocyte goes to osteoblast. And I mentioned another protein, RANKS2. RANKS2 is also responsible for lowering of the formation of osteoclast. It does that by increasing a protein called osteoprotogenin, that is a RANK-L receptor, receptor decoy. So it binds with RANK-L and prevents RANK-L from activating osteoclast. So all together, when melatonin binds to MT2 receptor, it promotes the osteoblast formation in mesenchymal stem cells. So the beneficiary effect of melatonin in bone health is well established. Now if I go to the next picture, so we already discussed how melatonin switched the conversion of mesenchymal stem cells from adipocytes to osteoblast lineage. So what we are trying to understand, since muscle and bone, myocyte and osteocytes are very closely related as they share the same anatomic neighborhood, what melatonin role could play in MSC to convert into a myogenic lineage? Because as I discussed earlier, in sarcopenia we saw that fat infiltration is one of the major cause. So maybe melatonin could do something similarly to bone to switch the conversion of mesenchymal stem cells from adipogenic lineage to myogenic lineage. So that is one of the major focus of my project. Now, so far I have discussed receptor-mediated action of melatonin, where it shows its activity on the differentiation of mesenchymal stem cells. But melatonin is also widely known as a potent anti-inflammatory. And here I have illustrated the mechanism of melatonin as an anti-inflammatory agent in bone. So when inflammation occurs due to oxidative stress, lot of reactive oxygen and nitrogen species are produced. They promotes the growth of osteoclasts. They disrupts the circadian rhythm. They inhibit the formation of osteoclast or new bone formation. Also, the inflammatory markers like NF-kappa beta, IL-6, and TNF-alpha disrupts the balance of osteoclast and osteoblast. So chronic inflammation will cause bone loss. How melatonin helps in here is that melatonin itself can bind with this directly with this reactive oxygen and nitrogen species because of its unique chemical structure, and indirectly it increases the natural antioxidant like sodium, SOD, glutathione, and also it increases the level of SIRT1 and SIRT3. Those are also function as an anti-inflammatory in mitochondria. So melatonin, by directly binding with the reactive species, lowers inflammation in the body, and indirectly by activating SIRT1 and SIRT3 and increasing the level of natural antioxidant reduces inflammation. So that's another beneficiary effect of melatonin, which is receptor independent, that helps in bone muscle. And as I also mentioned earlier, in sarcopenia, another major cause of muscle loss is inflammation. So both lipid infiltration and inflammation, in those cases melatonin can play a significant role to cure sarcopenia. But what already has been found with melatonin. So far, the literature showed that when given exogenous melatonin to the sarcopenic patient, it increased the myofiber size, it increased mitochondrial fusion, Christie prevention, and increased the satellite cell in the muscle cell that in turn cause more muscle growth. And inversely, it also inhibits the production of megamitochondria. Megamitochondria is an aberrant form of mitochondria that does not produce energy. It also reduces reactive oxygen species production, as we have mentioned in our last slide, and thus it reduces inflammation and also the apoptotic pathway. These are the role of exogenous melatonin that has been shown in different trial by far. Other than melatonin, so far I have been talking about melatonin and muscle. So I am bringing in another factor, which is exercise. So for sarcopenic patient, other than exogenous melatonin, exercise also seems to be very beneficial. In fact, exercise is the first suggestion that doctor will give to a patient who is prone to sarcopenia. Exercise actually controls the whole body's muscle health throughout our lifetime. It is not just a mechanical stimulation that anyone who exercise have on their muscle and bone. If a person go through moderate exercise throughout their life, or for say 3 to 6 months, there will be some molecular change in their body. So what happens when physical activity or exercise increase? It increases the insulin sensitivity and glucose uptake of the body. It also increases the number of satellite cells that will promote muscle growth. It reduces the inflammatory cytokines, and when physical activity occurs at a cellular level, it forms two different types of protein. One is PGC1 alpha that I will discuss later in my presentation. So that PGC1 alpha will increase the number of mitochondria in the body, and another protein they produce is mitochondrial fat binding protein, which will utilize the fat in the tissue to produce energy when exercise. Also with exercise, there is a greater demand of oxygen in the muscle cells, so the muscle capillary will increase. That will overall make the muscle health towards a better direction. So far till this date, for sarcopenic patient, good protein-rich diet and exercise have been prescribed widely due to this beneficial effect. Now, what exercise and melatonin does to each other, like how these two are related. In a study, it was shown that based on the intensity of exercise, the peak melatonin level is increased from the baseline. So this is a baseline melatonin level. So it is 24-hour time on the x-axis, and this is on y-axis. We can see the peak melatonin level. So this is from 10 a.m. to 10 a.m. next day. The black bar here represents the hour of darkness. So melatonin rise, melatonin level in peaks when in the hour of darkness. So here in the second graph, it's a low intensity exercise. The circle here, it represents the exercise time. And this is the high intensity exercise graph. As you can see, with exercise intensity, the peak melatonin level sharply rise from the baseline. So a part of my project is a clinical trial where we are investigating the similar efficacy of exercise, similar effect of exercise on the melatonin peak level. More on that I will discuss later. So relationship between exercise and melatonin exists. Melatonin is modulated by the exercise one person does, and it has been also proposed that due to strenuous exercise, lots of reactive oxygen species is generated, and as melatonin acts as an anti-inflammatory mediator, it can be beneficial to mitigate those inflammation and maintain the muscle and bone health. Now, what I am trying to understand throughout my project is I have mentioned total of 3 factors. One is melatonin, another is muscle health or sarcopenia, and the last one was exercise. So for my project, I will try to look into the role of melatonin in muscle health, like how it modulates myogenesis, how it shifts the mesenchymal stem cells differentiation to myogenic lineage. That is a part of my one of my project's aim. I will also try to look into the role of exercise on muscle health and properties. And along with all these, I will also try to see what exercise and melatonin does to each other and how all of these is baked into muscle function and musculoskeletal health. So this is the general goal of my project. So the key question I will try to understand is how do melatonin and exercise affect myogenesis and musculoskeletal health. My central hypothesis is melatonin and exercise improve musculoskeletal health and quality of life through their action on myoblasts and osteoblast depreciation and reduction in inflammation. These are my general aims. So my aim 1 is the preclinical study that I will be discussing in depth today. My aim 2 is in vitro study where I will determine melatonin effect on myoblast viability and differentiation on different cell type. And my aim 3 is the clinical trial where the impact of muscle strengthening exercise will be assessed on musculoskeletal health, circadian parameter, and mental health aspect of it. So for my aim 1, we try to identify the novel molecular targets. The LNC RNA is the target of our aim 1. We wanted to see what melatonin, exogenous melatonin does to those LNC RNA at LNC RNA after treatment of 1 year. So our goal is to see the changes melatonin bring at the genomic level. For my aim 1, I will refer to 2 animal models. One is a HER2-neu mice model on which the LNC RNA analysis was performed. This animal model received high dose of exogenous melatonin and then was used for LNC RNA analysis. And another animal model I will refer to is a ANAT knockout model where low dose of chronic endogenous melatonin was present. I will discuss the details in later. And those tissue from those animal model was used for Western blotting. So my aim 1 is divided into 2 parts. For my aim 1 day, like I said, we identified a novel molecular target or LNC RNA that was modulated by exogenous melatonin in HER2-neu female mouse bone. From that database, we try to identify the LNC RNA that are relevant to myogenesis and muscle health. In my aim 1B, we try to determine the impact of endogenous melatonin or the lack of it on protein or that regulates muscle function and myogenesis in a knockout mice through Western blot analysis. So for my aim 1A, the design was such that we wanted LNC RNA, we wanted to identify the LNC RNA that are modulated by exogenous melatonin treatment. The reason we went after LNC RNA was that it has been coming, like lot of papers are coming out, and in lot of research it has been shown that LNC RNA, although it does not have a protein coding ability, it performs as a regulatory modulator in the cells. It is responsible for many disease progression. Also, it is responsible for inhibiting many disease to progress. So we just wanted to identify like what melatonin does at a genomic level through LNC RNA to understand a novel pathway to better understand the muscle health and bone health of it. So what we did here was we so this is the HER2/neu mouse model, and this is the experimental design. So we divided the mouse HER2/neu mouse model. It was initially used for another experiment. So the mouse model was a breast cancer model, which was divided into two group, control and melatonin group. The control group received Faykill without melatonin from hour of darkness, and the melatonin group received melatonin 15 milligram per liter in the water they were drinking, which is equivalent to 6 milligram in human. And for 1 year every day the mouse were going through this treatment, and after 1 year the mice were sacrificed, and the bone from the female mice were sent to the bone of female mice was then analyzed for LNC RNA. And we have now in our hand this vast database of pathway analysis, co-analysis, and hierarchical clustering analysis, where we can see at the genomic level what LNC RNA did to this mouse bone. So before going into the analysis, I would like to go into the methodology a bit. So what we did after sacrificing these mice, we isolated the bone from these mice, and then it was RNA was extracted through using triazole reagent. Triazole is a reagent that is mainly composed of phenol and guanethioline, if I am not making any mistake. So what triazole does is during centrifugation or isolation process, it keep the RNA structure intact. So this is the figure where you can see that this is the red one is triazole reagent, and this is a pacemaker gel that we divide the RNA and the organic phase, and then the RNA of this bone sample was isolated, and then it was sent to Arista for LNC RNA analysis, and the rest was done by them. So I have been talking about LNC RNA a lot. So before going into the database, I'd like to briefly shed light on what LNC RNA is. So I think we all are aware that not all RNA codes for protein. Messenger RNA is the main one that codes for protein, and we have a lot of non-coding RNA in our cell. So there are two types of non-coding RNA. One is housekeeping non-coding RNA, which is transfer RNA and rRNA, and then there is regulatory non-coding RNA. And one of the major regulating non-coding RNA is LNC RNA. They are more than 200 nucleotides long, and they even though they do not code protein, they perform a wide range of function in the cell. In the nucleus, they interact with chromatin. They guide the transcription factor to the loci to promote the DNA expression in nucleus, and in the cytoplasm, it acts as a scaffold for mRNA analysis. It modulates mRNA splicing and its stability. It also scavenges out microRNA that may interfere with mRNA and protein synthesis. It also directly interacts with protein, sometimes as an assembler and sometimes as a decoy, preventing the assembly of two proteins. So in short, LNC RNA, even though it does not code protein, it has a wide range of function in our cell. So Now back to the result we got from Arrestar. So the LNCRNA that were upregulated or downregulated due to melatonin treatment of 1 year is presented here. So more than 3,000 LNCRNA were upregulated and 2,400 downregulated LNCRNA were in the melatonin treated group. So this is a volcano plot that represents the RNA. We took the log 2-fold changes in the LNCRNA expression. The green represent the downregulated RNA and the red represent the upregulated LNCRNA, and the black are in the middle are the one that was not affected by the melatonin treatment. Similarly, we also have the mRNA database at our hand too. So if we organize this LNCRNA in a Venn diagram, we find that around 2,400 LNCRNA were uniquely upregulated. Now, in our previous slides, we saw that LNCRNA and mRNA modulate each other. We want to identify the uniquely expressed LNCRNA that has like no interaction with the mRNA, which were upregulated for our study to identify what it does to muscle and bone. So then we did a pathway analysis. It is a computational biology method where the gene that we have identified, like upregulated and downregulated, was gone through a whole lot of online database to find out the pathway that gene overrepresent. Sometimes some gene will overrepresent multiple pathway. The relevant ones with our experimental design are presented here. So to explain the graph here, this is the upregulated LNCRNA and this is the downregulated LNCRNA pathway here. The dot size here represent the number of genes that are in that group. So the larger the dot means the more genes are present on that group, and the color of the dots represent the statistical significance. The more red it is, the more statistically significant the result is, and vice versa. So here I have marked the pathway that we will look into that is relevant to muscle health and myogenesis. To name a few is taurine, hypertaurine metabolism, signaling path of regulating pluripotential stem cell, JAK-STAT signaling pathway, fluid shear, circadian entrainment, and cholesterol metabolism. Initially, we will look into the upregulated pathway, and from downregulated, we will look into thermogenesis and oxidative phosphorylation. So for my aim 1B, before going into my aim 1B, we wanted to know that the data we receive from Arrestar, does it actually translate into protein or not, or can we actually take the upregulation and downregulation as a valid result? So the RNA portion was sent to Arrestar for LNCRNA analysis. In our lab, we had DNA and protein sample. So on those protein sample, we performed Western blotting to identify whether the up or down regulation in the LNCRNA translate into protein. We chose P per gamma as our target protein because P per gamma has came up through a lot of our analysis, and it is also relevant, as we saw earlier, in the mesenchymal stem cell switch from osteogenesis to adipogenesis. So in our Western blotting, we performed one-tail details. Here is the statistical analysis and it's a representative plot here. So when we see in the female bone that when you compare the P per gamma level in control versus melatonin, there is a marked decrease in the melatonin treated group in the level of P per gamma. This result also is in accordance with the previous finding of our lab, where we showed that when melatonin binds with MT2 receptor, it decreased the level of P per gamma and make a shift from adipocyte to osteoblast production. So this, and also in our LNC RNA, we saw the LNC RNA that that correspond to P per gamma also downregulated. So the data we received from LNC RNA is like we can go with this data forward. Now these are the LNC RNA for muscle. From those more than 3,000 LNC RNA we analyzed, we shortlisted these 4 initially to study the muscle health and myogenesis. The ones mentioned here are relevant to myogenesis and musculoskeletal health, and these are the downstream protein markers that they modulate to exert their function. So the one that I have chosen is MEG3, TAG1, MALAT1, and MAR1. As you can see that MEG3 was downregulated and it corresponds function to PPAR gamma. The protein that I have highlighted in yellow is already tested through Western blot in a different animal model, which I will discuss now. So before I jump into aim 1B, the future direction of my aim 1A is we would also like to assess the LNC, RNA and the protein relationship again in vitro. So we will probably in future we will probably run some assay where we'll use LNC RNA mimics or LNC RNA inhibitor to test those protein levels on the cells. And also we are trying to identify the microRNA that may be relevant to this LNC RNA function to understand the signaling pathway. So this is still very new, and we haven't decided it fully. We just have an idea. So this is a future direction for AIM-1A, and hopefully in my next seminar I will be able to share more details of it. So a few limitation of my AIM-1A is the female mice were only analyzed for LNC RNA, so we won't be able to assess the sex differences effect of melatonin in the LNC RNA, and it was a breast cancer model. So maybe the mutation that was introduced could have some effect on the LNC RNA analysis, but however, the mice were treated for 1 year with endogenous melatonin, and the data we received was very valuable to understand the effect of melatonin. So for my aim B, I wanted to determine the impact of lack of endogenous melatonin on protein regulating the muscle function and myogenesis. And the protein we selected were corresponding to the LNC RNA that we have discussed previously. The expected outcome of this aim was to see the protein level corresponding to the LNC RNA level we saw in the LNC RNA analysis, like the myogenesis marker will go up or down based on the LNC RNA's up or down regulation. So for this AIM-1B, we use a different mouse model, which is the ANAT knockout mouse model. ANAT is the enzyme that is responsible for converting serotonin and its downstream marker to melatonin. So in this knockout mice, the portion that is genetic portion that is responsible for ANAT production was deleted through CRISPR-Cas9. So the model could not produce the ANAT enzyme, thus melatonin production was depleted. So in these mice, we have absence of melatonin, and we wanted to see what it does to the downstream protein markers. So the first protein we tested was PPAR gamma in the skeletal muscle of this ANAT knockout mice. Here you can see that when we compared wild type versus knockout, in the knockout mice model, there is an increase in PPAR gamma level, which was found statistically significant. When we stratified the data based on male and female, as you can see, the relationship was lost in the female mice, but in the male, there was still a trend where knockout mice has more PPAR gamma level as compared to wild type. The reason is that we discussed earlier that melatonin is responsible for decreasing or inhibiting PPAR gamma activity. So in the absence of it, we expected the level would go up, and that's what it happened. And in muscle, PPAR gamma is responsible for energy production and insulin regulation, which it does by forming white adipose tissue. So the result we have is in accordance with the hypothesis we had going in. The next protein we analyzed was SIRT1, because SIRT1 was another major protein that is modulated by melatonin, and also it corresponds to one of the LNC RNA we found upregulating. So in the here, when we compared the wild type versus knockout mice, there was no statistically significant found in wild type versus knockout mice. But when we stratified it based on male and female, in male we showed, although not statistically significant, we found a trend that SART1 level is lower in the knockout mice as compared to wild type. Again, it's a melatonin depleted model. In knockout mice, there is lack of melatonin, and melatonin mainly function as an anti-inflammatory. One of the pathways that melatonin function as an anti-inflammatory is through SART1. So, in the absence of melatonin, SART1 level would go down. The next protein and another interesting thing that I would like to point out that other than melatonin, PPAR gamma and SART1 also co-modulate each other, meaning SART1 upon releasing it through AMPK pathway, it inhibits lipid synthesis and PPAR gamma function, and similarly PPAR gamma also inhibits SART1 and AMPK activity. So these two are always in an opposite relationship to one another, which is in accordance to our finding as well. The next protein we analyzed was RUNX2 in the skeletal muscle. Although there was no statistical significance found, there was a downward trend in the knockout mice, but the trend was lost in female mice and it remained in the male mice. RUNX2 level is also in an inverse relationship with PPAR gamma, and it is also modulated by melatonin in the bones. So RUNX2 is responsible for osteogenesis through production of osteoprotogerin, and this is just a supporting figure where we can where a test was done in vitro in osteogenic differential medium, where melatonin increased the osteogenesis through RUNX2. So this is another finding where we saw that in the absence of melatonin, the level of RUNX2 in knockout mice went down. So far, these are the three proteins that I have tested, and we wanted to see what the absence of melatonin does to these proteins that are responsible for regulation of adipogenesis and inflammation in the muscle. For my future direction, I will also see the protein level of CABP alpha, myopathy, and WNT5A that also came up in our LNC RNA and relevant to myogenesis and muscle health. I will also try to identify the same protein level on a different animal model, which is sarcopenic model that we have collected, and hopefully I will share the result with you in future. So the limitation of my M1B is it's a different mouse model than the hard-to new mouse model used for LNC RNA analysis. So there could be some interspecies variability, and the sample size for the female knockout was only NO2, where we lost some power for analysis, but we're trying to get our hands on more female knockout mouse tissues to get over this limitation. So the remaining aim 2 and aim 3 I won't be discussing today. To go over this briefly, my aim 2 it's an in vitro study where I will see the effect of melatonin on myoblast viability and differentiation. I will assess them on healthy myoblasts and also tissue taken from type 2 skeletal muscle. As I have mentioned before, that skeletal muscle is closely related to metabolic disorder like obesity and type 2. So, and also in this model, the inflammation type 2 diabetic model, the inflammation marker is high. So we will be able to assess the role of melatonin more profoundly in these tissue samples. And my aim 3 is a clinical trial where we will be determining the muscle strengthening exercise on the musculoskeletal health, what it does to melatonin level and circadian parameters, and overall the quality of life. I think the mental health aspect of our clinical trial is one of the unique things, because so far I have talked about what melatonin does physiologically, muscle health and all that, but all of these impacts mental health of a person, and it is also related to person's well-being. So we are also looking into that, like how exercise and melatonin affects the mental health of that person. So in summary, melatonin may promote myogenesis and reduce inflammation, improving musculoskeletal health and quality of life using in vitro, and we will try to show that using in vitro, preclinical and clinical studies. So before concluding my presentation, I would like to thank the past lab member whose work helped me to shape my project, my committee members whose support I have been needing very much throughout my PhD journey, the past collaborator and the funding of my present study. So thank you. I will be taking questions.
model, we did not make that
Yeah,
model.
we did not make that model.
That's from another web,
We
so,
collected this from.
yeah, it should have been cited on there.
Yes, yeah.
Hi, that was nice talk, and I'm kind of understand when you share the people gamma expression level in the try to extract the sample, so the level went down. So what we might interested in is like if the pathway goes to the osteoblastogenesis side, then we might see the different a change in the lungs to
Yeah,
level. So did
yeah,
you test?
we will test other relevant protein on the sample as well. The thing is, the triazole extracted sample, we have a practical limitation. The amount left is
Right,
not
right.
very much, so we are being very careful about which protein we want to test, because we can't as opposed to AN tissue, we have the whole leg tissue, so we can test a wide range of protein, go 20 microliter each and run many gels. But with this sample, we have to be very careful. We had a hunch that PPAR gamma is very relevant with myogenesis and the androgenesis and myogenesis switch. So that's why we went for PPAR gamma first, but yes,
Okay.
we will test the other relevant proteins in the future for sure. Yes, luckily.
Which cell type has the highest PPAR gamma? Is that what's? Yeah.
Usually
This was
adipose.
bone, right?
This is
Adipose.
mesenchymal stem cells, and in mesenchymal stem cells, if PPAR gamma is present, it will convert the switch to convert the differentiation in adipocyte direction as opposed to osteogenesis. But PPAR gamma is mainly present to regulate fats in the body.
So it's not, so it's hard to say, when you do bulk sequencing, it's hard to identify which cell type the change occurred in. But you think it's in the mesenchymal stem cells, this particular reduction is mostly in the mesenchymal stem cells?
It is also present in bone, liver, and skeletal muscle as well. But this study was done on mesenchymal stem cells, and this Western blot was performed on bone
Oh, I
tissue.
didn't realize that. Okay. So this is only on mesenchymal stem cells, it's not on some sort of whole tissue?
It's,
The one on the left is on whole tissue.
This is
What
a-
she's asking is what cell, how do we know it's a stem cell? Because they're bone cells, right?
Yeah, this test and this analysis was done on HER2 mouse bone model, bone cells.
So all of the bone cells, or all of the bone cells, so the
Yeah.
osteoblasts,
Yes, everything.
osteocytes? Okay. So PPAR gamma might be in those other cells too.
Yes.
Okay. But overall, there's no loss. It's just hard to know which cell type to attribute
Yes,
it to.
because all are mixed in together.
Yeah.
Yes.
Yeah, the model you used, the breast cancer model. Do you know what's the effect of that breast cancer on the bone? Is it changing your result or something like that, or?
I can't conclude for sure, but on the top of my head, for breast cancer model, many of the inflammatory marker and the pathway will go up. So on the bone, I think we also saw in our LNCRNA analysis, some inflammatory pathway and signaling mechanism went up. So I think that could be a contribution to the breast cancer model.
I think, but you wouldn't know this.
But yeah,
This
I know.
model is a breast cancer model where the metastases occurs to the lungs. So there is no bone marrow on the lung mets, on the metastases.
But we don't know what effects can have.
Oh, sure.
This cancer can have effect all over the body.
Oh, for sure,
Yeah, so-
especially on bone.
so-
But in this
You-
model,
yeah.
it does not. Yeah.
This one more question.
Yeah, please.
On slide 25, you've shown, slide 34. Western blot, there is a difference in the wild type male mouse. Like wild type, you can see the band on the two wild type ones, not the last ones, the female one, male one.
This one? Oh, wild type male.
Yeah,
This one and this one.
there are three.
Yeah, there are two.
The bands are two. Like, is there any
Like,
absence of the band in the male wild type?
No. So far in the wild type we saw like good bands for rounds too. But for knockout the bands are quite thick.
No, I'm comparing there are three wild type male on the right hand side.
Oh yeah, there is.
But the bands are only two.
Yeah, this one is quite thick. This. Just to compare, like this is.
What's the reason? Like all are wild type, but you don't get the band as good as like.
Yeah,
it was
the good
there.
as like the last one. Yeah, I understand
Yeah,
what
yeah.
you're saying. Like the wild type, the last two has a very prominent band, whereas the third one, there is no prominence. Yeah, I understand.
Yeah, like it's a massive difference. Like it's not like the lighter one in the.
Yeah, I know. I understand.
I have some questions.
Yeah.
I see several things here. One is that the exact, maybe that was a different one. That was a different slide that I saw, that was a sex difference that was interesting. For here, I think just because it's not significant in the females doesn't mean there's no drop in the females. It still looks like the patterns are very similar across the sexes, it's just a matter of how. Right? I mean, the females look a bit higher than the males, I guess. But overall, the patterns are the same, I think.
Yeah, do you think it's an N thing or a sex
I
difference?
think
'Cause I'd have the
I
same
think
question.
it's like we do see the similar trend for female as like the male, but yeah, we cannot control it for sure because, like I mentioned, like if we had more knockout, we would have found something statistically.
Yeah.
But yeah, I agree. There is a trend. Like what we see when we put all the male and female together, and when stratified by sexes, we see the relationship remaining.
Can I just elaborate on that
Yeah.
before you go? So, can you go to your cert one slide? Because
This is the
kind
one where I
yeah,
thought females were much higher.
that's what I was gonna say too, and I said, because you show a difference. A decrease in cert one in the knockouts implies that melatonin's increasing cert one in the
Yeah.
males.
Yeah.
And you were saying that it wasn't happening in females, but again, you're limited by an N
Yeah, no,
two.
yeah.
So, but I'm just gonna throw it out there, is there a sex hormone difference between the species regarding CERT1 regulation? And would you expect that?
Yeah, there could be. I think before just putting everything on statistical significance, I should look into biologically there. Because, not towards CERT1, but on the top of my head, melatonin does have different effect based on different sexes. So, because CERT1 is strongly modulated by melatonin, it would have different effect in male and female.
Like, do you know if there's a testosterone androgen interaction with melatonin in male muscle
There
versus
is-
female?
Yeah, there is like the melatonin level drastically drops in the female mice, which is in relationship with estrogen, and then in the male, due to testosterone, the level remains like the decrease is not drastic. So there is a relationship between melatonin with the estrus hormone, but I have to look into CERT1 how it plays into the whole sex hormone stuff.
But in this model, mice don't go through menopause, right? They're both intact.
Yeah.
So it's probably possibly could be a hormone or an N thing, so we just don't know,
Yeah,
right?
we just don't know.
I don't know if I asked this question last year as well, but then will diet affect this model? Like will diet, differential diet affect the PPAR expression or the different
Yes, definitely.
because I believe in real time you won't be able to control someone's diet, right? And I'm assuming most of the studies that you did was on a control. They were all on like a common diet, right?
Yes.
So, be it
so-
exercise or melatonin, will diet as a third variable like affect the entire
Definitely.
arrangement?
So to answer this question in real time, diet affects these protein levels drastically in our body. Like if a person is chronically on high fat diet, it will have more people gamma activity, and it will stunt the myogenesis and osteogenesis and produce more and more fat cells. So all the proteins related to those process of myogenesis, adipogenesis, osteogenesis will shift drastically. On the other hand, if a person is on protein-rich diet, it will make more bone and muscle. So melatonin will, so the body will produce more of those proteins that are related to myogenesis and osteogenesis. And diet is also very, like diet and melatonin is also very closely related. Like our carbohydrate metabolism and lipid metabolism is controlled by the secretion of melatonin. Like at night time we metabolize lipid, at the morning time we metabolize carbohydrate more, and that is all like tying together with melatonin secretion in the body. So yes, diet has a profound effect on the level of this protein and melatonin secretion. Yes.
I have a few questions about the ANAT knockout. This is a linear knockout model, right? So it's knocked out not specifically in one cell type, but everywhere.
Yes.
So does ANAT do anything outside of the pineal gland that might influence your findings? And where along the pathway is the ANAT? I think you might have that slide somewhere.
Yeah, I have it in. I think this is the synthesis of melatonin. So from serotonin to N-acetylserotonin, a precursor of melatonin. For this stage, the ANAT enzyme is required, and this is when this is blocked, the melatonin production is hindered. So this is the ANAT, and this is the more details metabolic pathway.
Yeah, I just feel like that might be affecting a lot of different tissues because serotonin is really very much expressed, not expressed, but synthesized at high levels in the gut and in other brain areas as well. So there might be effects that are extra melatonin, you know, like outside of melatonin.
Yeah,
So it's not all because of reduction of melatonin, maybe. It's too bad they didn't knock out the second, what you're pointing at right now, because that's probably only expressed in the pineal gland where the melatonin is synthesized. You know what I mean? Like,
Yeah,
remember
I
when we
know, like
discussed
the
it,
ASMT knockout would be.
Yeah, maybe they just can't, maybe it's embryonic lethal or something like that.
Can I piggyback off your question? So do you know anything about serotonin? Like, let's say you knock that out and there is like an increase in serotonin and tryptophan in the body. Do you know of any effects of those on muscle and bone and mood, hormone levels, anything that could impact your
Well,
research?
increased serotonin would definitely impact the mood, and but my understanding is like serotonin, if we just knock out this ANF, it won't convert to melatonin, but it will convert to other stuff and easily get excreted from the body. So
But serotonin is known to have bone effects, right?
Bone.
People on antidepressants,
people on antidepressant has more profound bone loss because
specific kinds of
specific
antidepressants,
kind,
right?
yeah. So yeah, if we have like an ANF model, we may observe the bone loss more profoundly because of the antidepressant effect or the availability of serotonin. That could be one thing that we should look into. Yes.
So you mentioned about exploring the effect of exercise on the action of or the relationship with it in
Yes.
terms of sarcopenia. I'm interested to know if you would be anticipating some effects on from the duration of, I mean, from the timing of exercise, like when it is done either in the day or midday
Yes.
or evening, and how will you account for that in designing your clinical trial later on?
Yeah, thank you for your question. So yes, without revealing much, we will be looking into that factor as well. Like how much, how many minutes, and when the person is exercising, what type of exercise, and everything. That is all factored in our clinical trial design.
Okay.
But is there anything we know about
Yeah,
exercise?
so from previous study, we know that the exercise done on the evening has more effect on the melatonin peak, like it causes a phase advance as opposed to if one person does an exercise in the morning, it causes a phase delay. So that type of study is done in a smaller scale earlier, and we will also look into that in our clinical trial as well.
Okay.
I have a question related to that a little bit. On your slide 14, you have your central hypothesis. So you were mentioning both melatonin and exercise. I'm wondering if you're, as far as your like expectations or your hypothesis regarding the two, would you expect them to have like an additive effect? Is what I was wondering, as far as, is it separate but then put together it's kind of increased by having both?
So we are not giving exogenous melatonin for this clinical trial. We would just measure the melatonin level in the urine and saliva of the participants, and we will try to see like that exercise effect and what it does to the melatonin level on those participants, and we will try to correlate these two. But yeah, since there is no exogenous melatonin, we won't be concluding something like that.
Well, it's 1 p.m. I think there's a chat question, and then I think we're gonna have to say, and go for the break, right?
This is a question from Dr. O'Donnell.
It's
I
okay,
answered
it's
that.
the same question.
No, it's actually, we answered that.
Yeah, so I think we're gonna say, let's take a break now for a
Stop
few
recording.
minutes.
No, I think I have another question.
Chetan.
Okay. So his first question is, in the animal study, are you using old age mice since sarcopenia is associated with aging? Are there any behavioral tests to evaluate sarcopenia in mice? So to answer the first question, the age of the mouse was, I think, around 14 months. So no, I'm not looking into the age aspect of sarcopenia in the ANAT. Rather, we are looking into the lack of melatonin into that model. And are there any behavioral tests? Yes, there are many behavioral tests in sarcopenia. It's I forgot the actual name of it, but it's like where the mice is made to climb through a specific wall, and in sarcopenic mice, due to loss of muscle, it's I forgot the test. I should know this, but yeah, I will get back to you in chat.
It's not the forced swim or the road.
So there is a sarcopenic model where the mice has to climb, and it's can't because lack of muscle and it's lack of strength. Yeah, so these, I will definitely get back to you with the behavioral test. So I think these are the two questions I have in chat.
Yeah, thank you, Oksana. We'll reconvene here in 5 minutes. Thank you.
So I will just stop there.
Did you stop the recording?
How do I stop
Chetan
the
would say.
recording?
Yes, it's that one. Yes. Yes.
Recording stopped.
This is much nicer in here. It's a much nicer. I think this is all new. How are you guys doing? I'm doing good. I have that big grant going on. That's why you didn't see me. I worked all day yesterday, so it's like, you would? And actually, believe it or not, we're doing so much with the clinical trial, like I pick them up, drive them back, keep transporting urine, so we're doing a lot of stuff with that, but yeah, I'm just, this little window of time to get it done, so it's going out for pre-review tomorrow. Yes. So. How's the work right out? Oh. But again, negative result isn't a bad result, right? Data are there, right? We're the truth seekers.
Most of the things are negative, like.
But that's okay. I wish we could, you know, just people who don't are publishing things that don't follow our hypothesis, because we're usually the more interesting findings, right?
The challenge is like since I'm not finding much in the associations with the
Yeah. When I was a graduate student, 9 months, I screened rabbit libraries for these muscarinic receptors, and I said, okay, I've had it. I need something else so I can graduate. It's like, I'm done. The Asana, give me quickly, that was really good. Good, good job.
I'm just not sure whether I should stop the Zoom meeting or not.
No, I think you have
No,
students
change
still.
from the Zoom.
Right.
We don't really, we just call it an exogenous dose. That's actually therapeutically relevant, because we're like, hey, that was introductory. Always make sure you say that. And then make sure you really know that paper.
I said mine not really.
Oh, so we have to.
I just emailed it to Dr. Lee.
I have
Oh, that's interesting, see? Oh, I didn't know that. Oh, that's good. Well, that's interesting. All right, bye, everybody.
Thank you.
There's so many lantern flies out there.
Yeah.
My,
Months, basically, they are on the road.
my daughter's, you know, an environmentalist, and she said that, you know, agriculturally, costing the state billions of dollars. But they said that after 2 years, they just left, because they were in central Pennsylvania. And now they're just gone.
Is
So-
it new over here?
Yeah, they came like,
I think it's
2 or 3
new.
years ago, so I'm hoping maybe they'll go away, because they're just squished all over the place, and they fly into your car, right, Jon?
Yes.
I literally opened the car to get them out of the Anyway, you guys have a good day. Actually, I think so. I have another meeting in College Holland. So I wanna stop here around 2:45. So I also, 1:45. Thanks. Anyway, I think that what we should do now is, Oksana, I'm gonna have you, gonna give you my questions during the committee meeting and via the feedback forms that we have. Unless somebody has like a burning question that they wanna ask, I'm gonna get started kind of just going through
Yeah.
the structure of a grant,
Yes, yes,
if that's all right.
yes, yes.
I have.
Go ahead, ask me while we're setting up.
Yeah.
Go ahead and ask.
Like,
For,
are you looking for basically total body fat in your study, as you are mentioning that it's the shifting off from adipocytes to
Are you asking whether I'm looking at for clinical trials or in my trial?
obviously like you can
Yes.
only look for your clinical
Yes,
trial.
we are looking that in clinical trial, like the fat percentage, the muscle mass gain, everything.
Like, do you think that with the use of the melatonin, the body fat basically decreases? Is there any study, because there might be something in the literature?
Again, since we are not using exogenous melatonin, we are just hoping that the exercise, like, we just want to see whether the exercise will decrease the fat level and increase the muscle mass in the body, and whether we can correlate that with the endogenous melatonin level or not. Like the spike in melatonin level, is it relevant to the gain of muscle and loss of fat or not? But since we are not
I'll be right
adding
back.
into any exogenous melatonin, we can't actually conclude that like, it's melatonin that's causing the pain.
But the you can,
Go ahead.
but the difference is like if you are only looking for the exercise, but how do you classify that it's only due to exercise or not due to the endogenous melatonin? That this effect is basically produced. The outcome is because of the endogenous melatonin production or the exercise basically. You know, like when you do exercise, you are saying there is a spike of melatonin.
Yes.
So the result is the effect of endogenous melatonin or from the exercise due to another route.
So the exercise we chose, one is classical and one is quite new. And one is supposed to increase the level of melatonin more than the others. So that is one control that we have. But yeah, to answer your question, we cannot directly conclude that from our clinical trial. That's why we're doing in vitro studies. And then somehow we try to tie this up. Like, hey, in vitro we saw the growth of muscle due to melatonin. And in our clinical trial, we also saw a spike in our melatonin level, and the patient also has a good muscle growth. So that's how we are planning that one.
But you can just add like the knocking out of production of the endogenous melatonin and doing the exercise then, and then looking at what the effect of exercise would be without melatonin.
Yes, in a
As a
animal
negative
model,
control.
you are talking about an animal model, right?
Yeah, in an animal model. Like,
Yeah, that
then
would
you
be-
can be like more sure or
Yes.
validate your
That's
research,
where
the study.
more, yeah, definitely.
That you can basically the melatonin is the effect is due to the melatonin, not due to the
Not exercise.
not due to the exercise.
Asset has a
Yeah,
good.
so when we're talking about for your clinical trials, so are you looking for like the change in total body fat?
Yes.
So but like if you're looking at total body fat for the entire body, like can it not be skewed because of like so sarcopenia and musculoskeletal diseases? Shouldn't it be better if you look for like specific changes in like the limbs of the human body?
We are also taking body circumference measurement along with the total fat percentage, so that is also there.
Well, if there's no more questions, I think we can kind of transition to the discussion of the grant structure for the comps and ORP. What I want you guys to download from Canvas, if you have your devices here, if you can share, is Manisha's comp exam. I just uploaded that this morning. I realized that wasn't on there in Canvas, but I think that we can discuss the structure of that. For some reason, my iPad is not updating this. Thank you.
Thank you, everyone.
Let's see what's gonna happen here. It's just not updating. That's fine. It might not work. Oh, it did work. Okay, so, I'm gonna let you guys directly see my notes on this because my other iPad is not updating, so I can't just read off my notes, from the other iPad. So here are my notes. So for Manisha's, comps grant, just a few things about the structure. First of all, she has an abstract, okay? So here's the abstract. Don't be too distracted by what I've written there. This abstract is followed by a public relevance statement that is also sometimes called a layman's narrative.
Yes.
And in the so for your comps, you don't only hand in just the specific aims page, the research plan. The students have also been handing in abstract and public relevance, okay? Now the abstract should be less than or equal to 30 lines. And there are formatting criteria that are written here. And this is all in the notes, if you download all of the notes from Canvas, 0.5-inch margins. You gotta stick to that even with your figures, because I got in trouble with NIH for like kind of scooting a little bit to the side there. No matter what font you use, and I like Times New Roman, you can't have more than 6 lines per vertical inch and 15 characters per horizontal inch. With Times New Roman, it's really cutting it close, but it's okay. Now, I like Times New Roman not only because I'm old and I'm used to seeing the serifs. You know, like for example, if you have a that's a pointer. If you have an I in Times New Roman, it'll be like that. You know, that's kind of what I'm used to, and it also squeezes more in. So that's another reason that I really like it. Okay, so 11 point or larger font, and again, Times New Roman, from what I've seen, is the best for squeezing in as much. And if you have a little bit denser font, then you can have more white space between the paragraphs, which I think is really helpful for the brain to have some white space. Because if you have, I don't know who had that, I think there was one person who had, there's very little white space in some of the proposals that the students give. That's very hard to kind of shift from one paragraph to another, put some space between paragraphs. Okay, okay, so you will get a grade for the ORP part of 1 through 6 based on NIH criteria. Yeah. Sorry about that. Should be 1 through 6 is passing, and then 7 through 9 for the ORP part is fail. So that's an additional way that the faculty kind of grade the ORP part of it. I shouldn't really call it ORP. I should call it the revised comps. Okay, and then the ORP is graded according to that feedback rubric that I've put in Canvas, which is a very good, solid rubric about like, does the student sound like they know what they're talking about? Are they defined to their advisor too much, and all that kind of stuff. Okay, so I wanted to make you guys aware of that. And what else do I want to mention here? Okay, so I am not really going to comment on the content of Manisha's presentation, her comprehensive exam at all. I'm just going to comment on the structure, okay? So that's that. And then let's talk a little bit about the aims page. Now, a lot of this you already know. I'm just driving it home for now. So the aims page is where the faculty and the reviewers will keep going back to. They just keep going back to that page, and so if something critical is missing from that page, they might miss it from the body of the proposal itself. So whatever is really important, make sure you put it in the aims page. I don't necessarily think it's important to write out how many millions of people are suffering from a condition. I think that's a little bit of wasted space. You want to talk about the scientific knowledge gap rather than this is a very expensive disease. Okay, because otherwise, if everything just depended upon how expensive a disease was, then lots of things would not be examined at all. It can't be scaling together with that. So it must have medical repercussions because, let me get my pointer, medical repercussions, because this is going to NIH and not the National Science Foundation. The National Institute of Health, as the words imply, they look at medical health issues. National Science Foundation looks at basic science that's not directly relevant to health, and the grants are usually much smaller. Okay, so mention the scientific knowledge gap up front, like don't just go straight into that, and then state the gap in the field, and that is really the most important part of your aims page next to the hypothesis, is what is the scientific knowledge gap that I'm trying to fill. And then you should highlight your hypothesis because they do want to kind of see that stand out a little bit. I like this particular hypothesis. I'm just going to mention one thing about this hypothesis, and that is that the student says essentially that a drug X or a process like mGlu R2/3 stimulation will lower a disease process Y. I don't know if you can read my terrible handwriting from very early this morning. So you are essentially the structure of a typical mechanistic hypothesis is that drug X lowers disease process Y by upregulating protein C. And that sounds so boring and formulaic, and it is, and that's kind of a very safe way to go. If you can think of something that's sort of more out of the box, that'll be very interesting, but this is very safe. And so make sure it's all verifiable. If some of the predictions that are in your hypothesis cannot be tested, don't include it in your hypothesis. Make your hypothesis very specific and less broad, because then that's easier to test and to verify. So all of that is, I mean, nobody always follows every single piece of advice when it comes to that. But if you have a big central hypothesis in the beginning and then more specific sub-hypotheses that kind of get to the nitty-gritty details, that's also okay. Okay, so, right, and so here, Manisha was approved to look at this project because it's not measles virus and it's not interferon gamma, right, Vivek? So, she's allowed to study this even though it's about virus. There's a virus component of it, and there's, probably some inflammation in there as well. I don't remember exactly, but it has to be, like, don't study, if you're doing breast cancer in the lab, don't make your comps on breast cancer. If you're doing melatonin in the lab, don't make your project on melatonin. And all that happens when you send your Ames page to the faculty is they approve the topic. They're not supposed to say anything about the content and whether they approve kind of what's in there. It's just the topic. And then you have another 4 weeks to hand in the real deal, like all the other pages, and with a modified Ames page usually by that time. And so that they're not like kind of giving the game away at that time. Okay. Now the two aims should not be interdependent, but they have to be complementary, and that's really hard. I struggle with that a lot. Go ahead, Asif.
So like if the specific aims doesn't get approved, so
That's usually, if the specific aims doesn't get approved, it just means the topic was not approved.
okay.
It doesn't mean anything about the scientific content.
But then does the 4 weeks time remain or
Yes. Well, no, you have to come back with a different topic, and that's a lot of work. What you really should do ahead of time is walk to the like all the professors' offices and say, this is kind of what I'm thinking, do you think this sounds reasonable, do you think you'll approve this topic, and then you write the Ames page, and then you submit it, and then you kind of have everybody's permission ahead of time. But this is like the formal part of that process. Okay, now not a lot of students do this. I've done this in the past. My very first NIH grant had one in vitro aim, one in vivo aim. It was very boring. It's like unless the in vitro can complement the in vivo and ask a different question, like the strength of in vitro studies are that you can look at different cell-to-cell interactions more easily, you can do a lot of mechanistic stuff more easily, so it's great to have that, but if it's like the same exact thing, like I remember my first grant, it's like the same exact question, in vitro, one in vivo, don't do that. It's not, it's like if you don't find it, I mean, what are you going to do with studies that say there's no protection in vitro, but there is protection in vivo? Then who cares about the in vitro stuff? It hasn't taught you anything different. So that's kind of one thing that I wanted to point out. Okay, now even if you're out, go ahead, Wilson.
If our both aims are in vivo,
If you're
both aims are
okay.
in vivo, and definitely if we are working on a disease in which there is only one approved model,
Okay.
and then both in
Yeah.
vivo study will have same model, same
Yes,
thing, just
but you can always ask different questions in the two aims still.
so is it fine to have both aims with
On the same model and the same protein or gene of interest, yes. But they have to ask different questions, like one can be, for example, what I've seen in grant proposals is one is like the short-term effect of this one drug on whatever process, disease process, and then the next aim is there also long-term protection against the disease process? That also is something.
Dr. Lee, is it mandatory to have an in vivo aid to the?
No, not at all. It can all be in vitro. Absolutely, totally fine. Okay, so here's the significance section. Now, I think it's very good to include a pathway diagram. You don't have to spend a lot of time on it. It just can be have the important words in there. You don't have to like make it really beautiful. Excuse me. But give us something, and again, the professors always come back to the aims page and they come back to the pathway diagram. And they always kind of scroll back to that and then ask you questions based on that. You'll see that a lot. And so the significance section here is nearly a page, and then the innovation section is pretty small. I have to say, for this group of faculty, innovation really is important. I'm just giving you that as an insider tip. Like, they're really into innovation. And so, I think I told you guys previously that at NIH, they say even if it's not perfectly novel, it can still be very important and it'll still get funded. But I think, like, from what I've seen, it's like, if it's been done before, and they look at previous literature to see if something's been done before, then that can kind of hurt your score a little bit.
If it's on the same, different model or different.
If it's on a different model, then it's innovative. If it's been done on a different model, it's highly innovative, this new model. Yeah, but don't copy like exactly what another study has done, not even word for word, but their complete structure of their paper. Just, hey, I'm in this other model, not in cancer, I'm in multiple sclerosis. Don't do that. Because we've seen that from students before, and I always call them out on it. Because you do kind of see what papers are cited repeatedly, and you go look at those papers, it's like, oh my God, it's exactly the same study from inhibitors to proteins of interest, everything was the same, and so that can be wrong.
Like, most of the time this just comes up like there's a latest thing in the market, and people are using it in every disease, and it's not in one of the disease. You can just pick up that and pick up that, and then
You could.
Does that, like,
That should
if
be
it's
fine.
a very close replica, like, does it validate the document?
It should not be a very close replica of another study that's out there, like almost everything will be the same.
Okay,
Yeah.
how do you differentiate significance and relevance?
Okay, you know what, I think they're both pretty much the same. Yeah, significance and relevance are the same thing. To me, physiological relevance is the significance. So you don't have to spend a lot of time or space on innovation, and I'd like to have bullet points for that. Here's 5 innovative points about the proposal or whatever. To make it easier for the reviewer to say, like, you gotta kind of allow the reviewer to scroll through fast and kind of pick up stuff like, oh, there were 5 innovative points, and then help them write their review. Okay, the approach section. I like how this is done. She has some white space, and then she writes that there's a specific, she repeats the specific aim 1 here, right? And then she follows with the rationale, or that nowadays is called the scientific premise. A few details about the experimental I'm so sorry. A few details about the electrode implantation, the behavioral seizures, and then she goes into, okay, specific aim 1A, the experimental design, expectations or outcomes, and then alternatives. So here, let's talk a little bit about that. We talked, I think, already in the past. There are biological controls, there are technical controls. What's a technical control? Just for the assay. Make sure the assays work. How would you do that on a Western blot? You would put the recombinant protein in one lane at the end and say, oh, my antibody recognized a recombinant protein, and the assay is okay. So you want the technical controls, maybe a positive and a negative control. You want more controls than you do in real life, because this is a grant proposal, right? And then the biological controls would be animals that have been treated with PBS, or animals that are wild type and knockout, etc. So you gotta have those in place, and you gotta spell out what they are for the professors. And you always have to have rationale for the doses you give, the frequency of the dosing, the timing of the dosing, the duration of the dosing. So the temporal dimension is really important when it comes to drug delivery, and often that's ignored. It's best not to just give one dose based on previous studies. Say you're going to do a dose response curve. Are they going to say you don't have enough money for that? Probably. But just like I recommend that you always do dose response curves, even though it's unaffordable. Okay, now the detail, so experimental design. How much detail do you want to put in there? All of the inputs should be detailed, but like you don't have to describe, I'm gonna run it on a gel, and then I'm gonna put it in a buffer, and then I'm gonna wash the buffer off, and then I'm gonna scan it on the eye. Like, don't do that. We do that in papers, but you don't do that in a grant proposal. But everything that you put into the running of the Western bot or whatever you're doing, all of those inputs need to be described. That's what's important, not how the Western bot is run, because we'll kind of accept that you know how to run the method.
It's like, is the, like, suppose like voltage and.
None of that, please. No, unless it's very unique. Like if you're doing a phosphorylation a gel shift, and like most of people don't know about it, then you can add some detail, which should be fine. Say, this is not standard, I've adapted it to our particular protein of interest, that should be okay. Okay, so in expectations and alternatives, I see a lot of people do this, including by people who are something around 10 age. Don't just reword the hypothesis. How does it move the field forward if the outcomes are positive? Okay, so that's important to have in there. And if the outcomes are negative, show that you could solve problems. So if it didn't work technically, how are you going to solve the technical issues? If it didn't work biologically, that is, the outcomes are truly negative and all the assays worked fine because you had all the positive assay control, the negative assay control, you had all of that in place, you know the assay worked, so the outcomes are truly negative and your drug does nothing. How does that move the field forward? Well, you can always say something like, now we know that protein X is not involved, something like that, like what will you still learn? We've still learned that protein X does such and such, but not so and so. That's what we've kind of been able to dissect apart. Okay, so you always want to give alternatives, right? So, like if my assay doesn't work, here are some alternatives I'm going to use. If my biological outcomes are all negative, here are some other genes of interest where I can manipulate those and see if there's an effect too. So that's also something you want to add as an alternative. Okay, this was kind of nice to have a treatment scheme to go back to. I thought that was useful. It does take up a lot of space though, so you have to kind of decide how you want to proceed. Have a future directions part where it's like it should be a logical extension of the current proposal, and like what is the long range vision for the lab. Like if everything's positive, what will we do? If the outcomes are largely negative, then how will we move forward? Either way, your lab has to move forward. It has to help your lab move forward and not just, well, we can't study melatonin anymore now. Like that. That's not going to be a good vision. Okay, now you need a section on rigor and statistics. This one, I have to tell you, it's pretty small and short. So I would expand on that part a little bit. What do you want to add? You want to add not just parts about the statistical tests that you'll be doing, but how are you going to do blinding, the rationales for your choices of dose, route, and timing. You can mention them here if you don't mention them in the aims itself. It probably belongs in the aim. Randomization, that's tough to do. Reporting missing data, like are you going to show all results, whether they're positive or negative? Are you going to include the outliers, or are you only going to show what they call clean data in PRISM, which cracks me up that that's called clean. But anyway, so you verified that the drug, and how will you verify that the drug reaches the tissue of interest and engages the target protein? Like you should verify that, especially if it's a new drug. Like if there's a lot of background literature that it gets into the brain and it binds to this blah, blah, blah receptor, then you can cite that work. But if you're looking at something new in a new tissue, like a drug has been used in the brain, but you're looking at bone, then you should make sure it engages the target and that it gets into the bone. So I don't know why, I think it's like historically, some student decided to add IOCUC and IRP stuff. Don't worry about it. That goes in other sections. This is like not a good use of space. What students should have done here is expand the section on statistics and rigor instead of IOCUC and IRB. Yes, we know you'll get IRB approval and you won't pick some people off the streets and say, hey, come in this room and I'll do some experiments. Okay, so why do grants sometimes not get funded? Now this is again all on campus, but let me cover it again. So in our little group, lack of novelty is like a big deal, okay? And if it's like diffuse and unfocused, like, okay, so this is hard because you're expected to really be, have lots of depth in your proposal as opposed to too much breadth, okay? That's kind of a subjective call, but try to look at what the other professors have said about the other proposals that are on campus. Make sure you read their feedback and kind of let that guide you. If the student does not, or the PI is not aware of some studies and doesn't cite them, then that is like a bad sign that they haven't really gone into the literature. And then, so that's so important. And then what I mean by no future directions here is that like after the outcomes are done and collected, it's a dead end. Like there's no clear segue into the next grant application. I'm guilty of that, I have to say, but anyway, not enough technical detail. Here with this set of faculty, you gotta have the technical detail. Not that I'm saying that you have to have the voltage of your apparatus while you're doing Western blotting, but technical detail as far as like, what are my negative controls for the assay? What are the positive controls for the assay? How do I know the assay is in the dynamic range of sensitivity? All of that needs to be included. Or I have to also say the professors really like to see time courses. In other words, and in real life, can you really do this? So you harvest your tissues, if you're doing an in vitro study and you're looking at kinase activation, you gotta do that boring time course. Like you gotta collect that 15 minutes, 30 minutes, 3 hours, 6 hours, 24 hours. It gets to be so much work that you have to propose it. Because otherwise it's like, well, what if the changes only occur in 3 hours and you've just collected at 24 hours? That's like not, for kinases, that just doesn't work because they shoot up and then they come back down. So you have to have the time course. For animal studies, do you want a time course? Yes. Why? Because most diseases are progressive, right? There's like an acute injury phase, there's a progressive injury phase, there's a subacute injury phase. You gotta have all your bases covered like that. Don't worry that it's $100,000 US, like just propose what makes sense to make a good story, okay? Like don't start to do a budget or anything like that. We've had students do that. Okay, so you want definitely a self-critical approach. In other words, I'm aware of the flaws and the limitations of our work. At least there's that, right? And here's how I interpret it in light of those potential confounds. So you want to have that as well. Okay. So in other words, you want to list how you'll interpret negative data and list some alternative pathways. And some of the parts of your proposal may have some risky aspects. You gotta just kind of manage those. Try to stray away from that for comps and ORP. It should not be overly high risk. It should just be like, a lot of it should be relatively straightforward and safe. So that's what I got for you. Yeah, do you have any questions? Yeah, Rachel.
One about the technical part, not having to necessarily go into all the nitty gritty with Western blotting. Similar to the models, as far as an in vitro model or an in vivo model, can you say as established in and cite have the kind of staple paper?
Yeah. To
that
save some
you're,
space.
instead of saying I'm going to do this, then this, then this.
Yeah, I mean, if the model has been around for 20 years, 30 years, state that. This model has been developed since 19 whatever.
Yeah.
And it is very well established that it leads to behavior deficits, histological changes, biochemical changes, and that's been studied, and that's already been like well established. Yeah, it's totally fine.
And if we are choosing a drug,
Yeah.
and it's being used for a single dose for a certain time,
Yeah,
and it's
right.
working.
Right.
Even then, we have to put
Yes.
a higher dose and a lower
Yeah,
dose.
because you're in a new model, remember? God would know. It's going to be the same dose. You
Yes,
don't
yes.
know. It's better to do a dose response curve, even if it's just 3 doses. Yeah.
Yeah, like I've heard that the faculty is more interested in drug response curve when they say you that what their
Yeah,
main question.
there's a reason that in pharmacology the dose response curve is like very well viewed very highly because like there's so much information to be gleaned from that. You know, you can't see efficacy unless you have a dose response curve. Yeah.
But on the other side, they can also ask why you are looking for a higher dose and lower dose if this is working.
They
Or,
could
like,
say that, and you could say because I'm in a new model and I don't know how it'll work out in my lab. And there's been literature that it's a little bit variable. You know, it's always going to be variable. You know. Yeah, no, I would go safe. I would do dose response curve for sure, and the temporal. Once you have a good efficacious dose, then I would do a time response curve. You know, over time, how like is it lasting protection or does the protection wane after 4 weeks? Like I think that's also an important factor. You okay, Oksana?
Yeah.
It's like after a presentation, who wants to sit for a class?
No.
Yeah. Well, that's all I have for you guys, and let's see what time it is. It's 1:39. I really hope you find the information on campus useful. So, and I will, next week is Muslim.
Yeah.
So, I need to kind of know from all of you what you guys are deciding to do your writing assignment from. Do you want to kind of go through the group? Have you done this before? I need to write it down. Let me just get some piece of paper here. So, because I want to know what to focus on in my lectures. So let's start with you, Jan. Did you want to do a grant or a literature review or?
So mini review about
Mini review.
mechano signaling regarding melatonin.
About, sorry, say that again?
Mechano signaling.
About what kind of thing?
Mechano.
Oh, mechanical signaling.
Yeah,
Got
yeah,
it.
yeah.
That's cool. Mechanical signaling in bone
Yes.
and muscle.
Yes, yes.
Bone.
On.
Okay. So it's just a literature review. Okay,
Yes.
that's fine. Asif, you are, I'll get to you in a second, Melanie. Asif, you want to do, I encourage you to do a specific aims page.
That's what I was like. Can I do my?
No. I'm just kidding. Go ahead and ask your question.
Like the specific aims I plan to do for my comps. Can I do that?
No, you can't. I can't give you feedback on it. It can't be from faculty. It has to be from peers. Yeah. So you have to find something else.
Either a specific aims or an introduction.
Or an intro to a paper or your dissertation or paper. Okay. All right, Asif, I don't like that particularly very much. I think you should try to do something. Maybe you want to do an approach section. Like an intro to a paper is very basic, right? I know you're really busy right now. We'll talk offline. Let's bug Melanie now.
I'm thinking of doing a literature review on protein secretion mechanisms.
That sounds very interesting. Protein secretion from what kind of cells? Any kind?
And we're looking in endothelial cells.
Got it. From endothelial cells. Got it. That sounds great for a first year kind of thing. Young, Asif, Melanie, Rachel, what would you like to do?
I was thinking like part of the results section for Cooperzone. I did the method section last time, so.
Yeah, that flows. That sounds like a great idea. You've got the figures for it. You want to hand in the figures too?
Yeah.
Or, yeah. Perfect. All right, Muslim.
Ma'am, ideally, like
Ideally, got it. Okay.
specific aims page,
Got it. Great.
but
Ideally, but
if-
I know what you've got going on, but still trying to get in the aims page. Yeah.
but if not, then something like literature review.
Okay. I mean, yeah. I think during your second year, okay.
Even methods.
Try for the aims page. I highly encourage that.
I will try my best.
Try your best.
You know, like it will take a lot
It
of time.
does. It does take a lot of time. It's true.
And most of
It's
the
true.
time
The aims
is
page is a killer, right? Rachel, are you alive? You survived it?
I did.
That's good. Specific aims page. Okay, we'll see if Muslim is still okay.
like,
By the time Christmas rolls
and it's not good to do work in ACF, you know, like
Yeah,
I will
yeah,
be
yeah, yeah.
spending a lot of time.
Right, I know what you mean. I know what you mean. Ah, gosh, this is the same situation that Asif is in, right? Yeah, I'm so sorry that I have to bug you guys with this, but I think, give it a shot, right? I mean, how much of your grade, so your oral presentation and the handling of questions is the majority of your grade. And then the writing assignment, I've reduced that, and I've expanded how much of your grade the participation is, okay? So that kind of reduces it makes it more low stakes rather than high stakes, so that you can have fun with it, play around, and like kind of just test your writing out and test your ideas out.
For colleague, I was thinking of expanding on my previous review, but then I don't know, like if you will be interested in assessing that again.
No, that'd be okay with
That's-
me.
It should be an expansion to what
Okay.
I did like in my first 2 semesters. But yeah, I don't know, like if you.
When will you take your comps? Like, I want everybody to think like 1 year out. If you want to take your comps next year, try to do the A's. I'm kind of changing that advice a little bit. I know you guys have a lot going on, but getting some practice down. Maybe we should do it like this. If you want to give me outlines to start with, and just kind of, even if you don't have to write out perfect English, let's look at the scientific ideas that you have proposed in the names page, for those of you who are going to take your comp soon. You know, let's do it that way, so that it's less kind of, because that's like the main thing, you know, I mean, anyway. Okay, so you think about whether you
Sure,
want to expand, or you could do both, that's also okay. If you
Yeah.
want to hand in like a skeleton
Right.
of a idea for a study, for a grant proposal, and also expand on your previous review where we kind of go into the writing more, that's okay. But I want to give you both, so I'll give you the writing feedback and the scientific feedback for the review, and then the scientific feedback for the specific games page, but not the language feedback.
Correction,
Okay?
Yes.
Yep.
So, right now I was thinking about doing a mini review on the types of exercise and its effect on melatonin level. So that was primary plan, but I will also think about the aim and the comp that you just mentioned.
Yeah, I mean, that just occurred to me. What do you guys think of that? Do you want to try that too? I mean, Vivek?
Like I can try giving the review plus the outline of my specific games.
Yeah, and an outline can be very skeletal, right? I mean, it would be the same like, for example, I mean, I know where you guys are coming from. You want to do the review so that you read up a lot of the literature and are kind of well versed in that. And then as you read the literature, you read more methods. And then those methods you can apply to your comps and ORP, because you gotta have some knowledge of the methods that you propose. A lot of the questions will be related to that. Let's all talk about that offline, okay? Because I have to go to this other meeting. Because as always, I don't want this class to be a burden. I want it to be helpful to you guys. And I know that writing a very like polished games page is going to take you weeks, right? So I want you guys to come visit me in my office and talk about it offline, especially the senior students like you, Rachel. You know, kind of what you've been through and what would have been helpful.